Hi everyone, today I'll be talking about my final project for SEAS, which is predicting mosquito habitats via land cover data. Let's get started on why it's important to conduct research with mosquitoes. Author Timothy Weingart says that statistical extrapolation situates mosquito-inflicted deaths approaching half of all humans that have ever lived, which is to say nearly 53 billion human lives. And each year we lose another annually millions of lives to mosquitoes. So mosquito research is important and is kind of untouched and underfunded right now. The goals of my study are to help scientists to be able to predict and track mosquito larval habitats, which can hopefully help predict vector-borne diseases and other vector populations. And personally, I'd like to explore new technologies in this project and just discover more about the topics that we're talking about right now. And what I did to conduct my research was, one, I analyzed the actual land cover of my community by taking several points across a wide distance. I hope to get a accurate representation of the land cover of the average land cover in my community. And I did so using the Collect Earth Online software, which you can see here. There's different land cover elements, which I label, and the results are pretty clear cut. Tree canopy cover seems to be in the most predominant land cover element, with cultivated vegetation and impervious surfaces coming in second and third. So this tells me that there's a lot of shade in my community and there's a lot of vegetation. So those are two important things to keep in mind, because when you're reviewing the literature, which I read a book, The Mosquito Human History of Our Deadliest Predator, and various different studies provided by my mentors and other online articles, I came to the conclusion that mosquito larval habitats must have a body of water which should be nutrient dense, be devoid of predators, and have a stable temperature. And they should have some sort of food source, but namely aquatic plants. But I didn't find something conclusive on the luminosity of the habitat, which I found interesting, because I had the intuition that shaded regions would be better for mosquito habitats. So in part 3, I acted on that. I had 4 different time periods, which are unshaded with tap water, unshaded with sugar water, shaded with tap water, and shaded with sugar water. And I placed them in 5 different areas: urban, grassy, lightly wooded, densely wooded, and near a larger body of water. And the results were pretty clear cut too. I saw that the shaded plus sugar water across the board had the highest number of mosquito larvae recorded. And generally you see the list, that's what the results looked like. Although for result number 3, the grassy trap did not experience the result of unshaded plus sugar water increasing mosquito larvae. So that leads me to the conclusion that the sun exposure is actually a very important characteristic in determining mosquito larvae. There has to be adequate shade so that their temperature does not fluctuate wildly, and so that the mosquitoes have a better chance of surviving. And the urban trap throughout this entire study did not receive any, I did not find any mosquito larvae at all, which is to be expected with all the impervious surfaces surrounding it. And a possible extension of the study, which I got thinking because of the temperature fluctuations, was using climate data to further enhance this model. So, for example, I found more mosquitoes towards the end towards the higher temperature days of my study. So perhaps there's some sort of correlation between that, and I think that merits further research.
Hi, I'm Marina, and this is my final research project. My project studied the 2012 West Nile virus outbreak in the United States, specifically focusing on Texas, who accounted for about 1/3 of the cases. There were 6 counties in Texas that reported over 50 cases. These counties included Dallas County and Travis County, the main focuses of my research. Most of the outbreak was focused in the central area of the United States, as you can see on the map below. The goal of this project was to find if there was a correlation between land cover and the West Nile virus outbreak that year. My research question is, how did land cover play a role in the 2012 West Nile virus outbreak in Texas? As I mentioned, I chose Travis County and Dallas County to study specifically. I chose Travis County because I live here and know a lot about the area, and because there was a high number of cases reported that year. I was also able to complete my own globe land cover observations. I chose Dallas County because it had the highest number of cases reported, almost 400, and because I was able to use another SEAS intern's land cover observations from this area. The top set of photos is an example of a land cover observation I did from Travis County. It was classified as MUC code 91, which is a residential area. The bottom set of photos is also a residential area done by another SEAS intern from Dallas County. These were both in the suburbs of the counties, away from the downtown areas. Comparing and contrasting the land cover observations helped me in determining the differences in Travis County and Dallas County land cover. Here are some more land cover examples for Travis County. The top set of photos was classified as MUC code 02. It was taken near a water plant in the back of my neighborhood. And the bottom set of photos was taken on a hike in the back of my neighborhood, and it was classified as MUC code 56. It's also important to note that the hike had running water at the end of the trail, and both of these sites actually had running water, which could be potential mosquito breeding habitats. These photos also show the diversity of land cover in Travis County. Here's another example of a land cover observation in Dallas County. Here we have a residential area next to the water. This means that there is potential mosquito breeding habitats and also a high possibility for people to get bitten. Here's a quick overview of the geographical factors that helped the virus spread that year. Local officials said that precipitation and above average temperatures helped mosquitoes thrive. Especially in August, as you can see, there's a peak in precipitation and temperatures are pretty high. There was a peak in the West Nile virus cases reported, where 225 cases were reported in 1 week. Here are also some graphs I used. This one on the left is soil moisture and the one on the right is vegetation index. All three of these graphs were collected by Climate Engine. I used Appier's land cover data to find the land cover of Travis County and Dallas County in 2012, and then I put that data into a pie chart. As you can see, the savannas, urban and built-up lands, and grasslands was the majority of Travis County, and Dallas County had more than half be urban and built-up lands, and the rest was mostly grasslands. These are both according to the land cover type 1 and the IDBP classification. In conclusion, Dallas County had a higher number of cases compared to Travis County because it has a higher percentage of urban land cover. Dallas County also has a larger population living in a smaller area, which means they have more compact layout compared to Travis County, which is more spread out. Urban development, especially the ones with nearby water sources, are a large part of the West Nile virus cases. Residential areas are a large concentration of people, and they have many sources of water, such as gardens and empty bottle caps and more. That could be potential mosquito breeding sites. People are very susceptible to being bitten in these areas. Especially suburban neighborhoods, they have housing integrated with vegetation that's cultivated and watered on a regular basis, which allows mosquitoes to thrive. A similar study I looked at stated that there were greater numbers of disease vectoring species associated with human habitations, so they found similar results. Here are the best features.
I'm Samuel, and along with my research partners Elizabeth, Bruchel, and Frank, we analyzed the environment and vegetation around mosquito habitats. Before we start, it's important to know some characteristics of mosquito habitats. These habitats tend to be secluded, dark, and near bodies of water for larvae to grow in and can be almost anywhere. This picture here is an example of mosquito larvae swimming in a bottle cap. So the goal of this study was to determine the relationship between a mosquito habitat and the environment and vegetation.
To begin, we contacted an official from the Department of Health in Chicago who was able to provide regional mosquito population statistics for 5 established mosquito habitats, with the majority had experienced a noticeable increase. Our group member located in the Chicago area then visited each site and recorded the land coverage types and percentages using NASA's Globe Observer app. We then cross-referenced our ground data with the satellite images from Google Earth to create a more comprehensive vegetation model for the area. Using ground cover percentages from Globe, we generated 5 separate pie charts reflecting each area's makeup of vegetation, and then compared the charts to the mosquito population data from the Department of Health to determine which area, and therefore which vegetation majority, could be linked to the increase in the number of mosquitoes. Pictured here are our 5 analyzed locations in the Chicago area.
So here were our findings. After we analyzed the data, we drew up pie charts for each site. Each habitat was ranked based on the percentage increase of mosquitoes in the area. So Habitat 1 had the greatest percentage of increase, and Habitat 5 had the smallest. Habitats 1 and 2 were situated next to a large body of fresh water, and they were dominated by deciduous trees and grass. This habitat favors floodwater mosquitoes, where eggs laid in the grass hatch when the water level rises and saturates the ground. We also believe deciduous trees to be a major factor in this sort of environment by supplying female mosquitoes with shelter, protection, and a source of sugar to feed on. Habitats 3 and 4 similarly were also dominated by deciduous trees and grass. However, unlike habitat types 1 and 2, they lacked a large body of fresh water. This data here essentially confirms the positive correlation between deciduous trees and grass and mosquito populations.
Lastly, habitat 5 actually decreased in mosquitoes. We believe this decrease to be attributed with the flowers present, which may contain certain compounds or natural volatile oils that repel mosquitoes. This data seems to show that certain flowers are very effective mosquito repellents. From analyzing our results, we were able to conclude that mosquito density increases with deciduous trees and grass, but the growth is limited with shrubs or other residential flowers, Fidelis and Yarrows, which are known to repel insects.
Hi, my name is Kavita, and I'm a Mosquito Mappers intern this summer at NASA. My hypothesis for my research was that mosquitoes are more likely to inhabit areas with high rates of poverty because many of the factors of poverty and the factors that attract mosquitoes overlap. My first research question was whether or not mosquitoes do inhabit areas of poverty, and if so, I wanted to understand why. This research does have large implications because if impoverished communities are more at risk to mosquito-borne diseases, this places minority communities at an increased risk, and this also dictates how governments and communities should allocate resources to minimize the spread to vulnerable communities. For this research, my data was collected from the CDC program, ARPANET, and aggregated from the US Census Bureau and the American Community Surveys. This analysis was all performed at a county level and started with building a linear regression model which took into account 5 variables: poverty rate, square miles of water, population, population density, and the average temperature per year. The first model output showed us that the multiple determination coefficient, or the R-squared value, was 0.73. This means that 73% of the total variations in the cases of mosquito-borne diseases are described in this model itself. And to further understand which component of the model contributed the most to the R-squared value, we ran individual linear regressions, and we found that population was the biggest contributor to the model. And this may be because urbanization creates habitats for mosquitoes, or it may be because there's a larger population to infect in cities. It was found that poverty rates do not necessarily impact the total number of mosquito-borne disease cases, which may be for a number of reasons, including the fact that poverty at a county level aggregation is not specific enough. It also may be because many of the poorest counties are rural, and they are not at risk of mosquito-borne diseases. On a regional analysis, it was found that the Southeast may suffer from a severe lack of data because the R-squared value was so low. There might also be another variable that must be considered for this region especially. On a further analysis between Cobb and DeKalb counties in Georgia, it was found that their major difference was their poverty rates. And the other difference was that Cobb had 5 cases of mosquito-borne diseases over the past 5 years, and DeKalb reported 11. And to further analyze these 2 counties, I mapped out the land cover elements using satellite data from Collect Earth Online and tried to understand the distribution. Cobb and DeKalb have very similar distributions of land cover elements. And as for poverty analysis, the land cover elements were mapped over a poverty map for both counties to better understand what land cover elements are present where. This is useful for future research with mosquito-borne diseases. And for future actions that we need to take, we need to integrate a GLOBE program in cities to track mosquito data at a very granular level. And this can be matched back to census tracts to create a very specific level of data to allow for a much more complete analysis. And an important part is using raw mosquito data to predict outbreaks rather than past outbreaks. And cities need to take steps to ensure that they have an integrated pest management system in place.
Hi, my name is Virginia Weston. I am on the Mosquito Mappers team for the CES 2020 summer internship. And I did my research project about the success and failure in finding mosquito habitats based on the land cover type and the site and the area. So for some context on mosquito habitats, Mosquito habitats are typically in an area with a lot of shade and vegetation that's far away from predators, a still body of water that's at a lower elevation. And based on these land cover types, this will influence how long the danger days are in areas. And danger days are basically just the days that are more likely to experience a mosquito vector-borne disease outbreak. So as the climate changes, mosquito behavior will also adapt, that's important to note. So for my project, I chose 2 sites to look at closely. One site was from my 3.5 kilometer grid that I made earlier this summer with Globe, and this site had no mosquito larvae. And the other site I found on the online Globe Observer data access tool and visualization system, and this site had a lot of larvae. So for site 1, this is information about it on Globe. There's no water source and no larva count. And from the ground, this is what the land cover photos look like. As you can see, it's a college campus with lots of tended grass and trees. So we call this cultivated vegetation because it's irrigated by humans. And then from the satellite, you can tell that there's a lot of cultivated vegetation, which is the pink color, and buildings, which is like the peach color. So compared to site 2, this site is near Minneapolis, and there's a water source recorded in Globe, and there was a larva count of 30 recorded in Globe. The land cover ground photos here look a little different. As you can see, that's a picture of the water source right here on the bottom. And then this we would call grass because it's less tamed and probably not irrigated by humans. So that's some differences with the land cover type. And as you can see from the satellite, there's not a lot of cultivated vegetation. There's more trees, grass, and impervious surface. So now we're going to look at the larger area of interest. This would be the 3.5 kilometer area of interest surrounding site 1, where there is no mosquitoes found. And the land cover type is mostly urban and built up lands. And as you can see over the course from 2000 to 2018, there's not a lot of change in the land cover type. And the elevation from the Aster satellite measured at about 317 meters. Compared to the site 2, where there was mosquitoes found, there's a similar land cover type. There's mostly urban and built up lands and not a lot of change over the course of the years. And the elevation is also similar. It's at 288 meters. So based on this information, we can tell that there's not a lot of difference in the area's land cover type, and this is most likely due to the fact that the elevation is pretty much the same and there's not a lot of change over time. But the microclimate where the sites were different. They had a different land cover type, and actually the college campus, because it's cultivated vegetation, it is also sprayed with pesticides against mosquitoes. So the microclimates are really what impacts the presence of mosquito habitats or not.
Hello, my name is Ryan.
My name is Gautam.
My name is Shantanu.
And I'm Emily. Together we are the Mosquito Mapper interns for the 2020 NASA SEAS internship.
I would like to start us off by introducing our topic. The introduction of satellites has revolutionized science in many ways, from exploring space to looking at our own planet. While satellites have provided us with lots of groundbreaking data, it is important not to overlook in-situ data. Not many research papers compare both in-situ data and satellite data, so we hope to do so that scientists can know which one is more efficient and feasible for their needs. We will compare the differences between in-situ data and satellite data for the same plot of land and display the differences in a statistical form.
So in our team of interns, which is about 100 or more people, each intern chose a plot of land near to them, and then these coordinates of these land plots were put into a tool called Collect Earth Online, which is a web-based GIS tool. And so this tool took the plot of land and created a grid of 121 points on that land. So then we could classify exactly what type of land cover is on that plot. So tree cover, water, grass. And so then the interns also took in-situ observations through the Globe Observer app, and then we downloaded these pictures from the Globe Data Visualizer to help compare the two. And so what we did was we took averages of the types of land cover, those percentages, and we compared them between satellite imagery and in-situ observations. And we made graphs to better visualize the stark differences between some categories.
Here are some of the things that our data has shown us. These are the pie charts for the satellite data and the in-situ data. We can already see from here that the biggest differences are in the categories tree canopy cover and cultivated vegetation. Most of the other sections seem roughly similar with a few differences here and there. These differences are highlighted in this bar graph, and it is easier for us to compare each category with each other. Not only do we see that there was more tree cover and less cultivated vegetation in satellite data, we can also see the less dramatic changes like in treated pools and shadows and unknown areas. We can quantify the changes more easily by looking at this table. We can easily tell that the satellite images had around 17.65% more tree coverage than in-situ images. However, the satellite images had 16.89% less cultivated vegetation.
So as we see in the graphs, the tree canopy cover percentage in the globe land cover photos is much smaller than the satellite data, and the percentage of cultivated vegetation and bare ground are greater. The biggest reason for this is that the globe land cover photos allow us to see beneath the tree canopy cover, revealing an entirely different perspective for the same plot of land. As we see here, the satellite imagery shows mostly tree canopy cover, but the in-situ globe land cover photo reveals a trimmed lawn underneath. We can also see the grass in front looks a bit more wild, while the grass in the back looks neatly trimmed. This means that the grass in the front is classified as grass, while the lawn is classified as cultivated vegetation. In another example shown here, the satellite image reveals what seems to be pavement, which should be classified as an impervious surface. However, the in-situ photo reveals that there's grass growing beneath the stones, which should change up the classification a bit. However, the Collect Earth Online satellite photos allowed us to see a much larger area. This is why we were able to identify a treated pool using the satellite data, whereas the same pool was not visible in the in-situ globe photos. This explains why there was such a large drop in treated pool and lake, pond, or container water.
After analyzing the results, we have concluded that neither method is better than the other. Satellite data can provide a more widespread view of the land, while in-situ data can provide a more detailed view of a smaller plot of land. These two methods should be used subjunctively to get a maximum analysis of the land. Future improvements to the methods could be having more citizen scientists to provide in-situ data, or have vehicles equipped with cameras to provide in-situ data.
Hi everyone, I'm Jasmine Wu, and I modeled Michigan's West Nile virus cases while also taking into account climate change's unique effect on the Great Lakes area. While I was collecting Globe Observer data, I noticed that many lawns were almost completely yellow, and I found no mosquito larvae in our cities, man-made lakes, or my traps. This inspired me to investigate the relationship between climate change and arboviruses. The West Nile virus is spread by Culex pipiens, which is shown on the right. Michigan is the 12th most cases in the U.S. and has experienced outbreaks every year since 2002, when West Nile was first introduced. Contracting West Nile virus can be serious, depending on if you're the 80% that's asymptomatic. 20% will experience fevers, aches, and vomiting, while 0.67% may experience comas, tremors, paralysis, and death. A model that could help predict future cases of West Nile can guide control efforts and allow health departments to prepare for outbreaks. Climate change has affected the Great Lakes region in a really unique way. As you can see here, average temperatures, precipitation, and dry days are all expected to increase. Michigan will experience hotter, drier summers and really wet winters, along with more extreme droughts and storms. This also results in extreme high and low water levels. Given these predictions, we should know how they will influence West Nile transmission. As temperature increases, mosquito and viruses mature and reproduce faster. However, after a certain threshold, mosquito mortality increases and populations go down. As precipitation increases, more potential mosquito habitats are created via puddles or sitting container water. However, excessive or violent rain can displace larvae and destroy mosquito eggs. It's important to find the right balance. Drought has also been found to increase West Nile virus transmission by forcing mosquitoes and birds together at limited water sources and decreasing mosquito predator populations. In 2012, one of Michigan's worst droughts, you can see a dramatic increase in cases. Additionally, in 2014, the Metro Detroit flood was a result of excessive rain, and as you can see there was only one case that year, tying back to what we found in the last slide. The peak at 2002 in red is so large because the population has not built up any herd immunity yet from previous infections. I did not include immunity in my model because it is difficult to measure accurately and I could not find any reliable data for it. Now I'll go over the process behind creating the model. I gathered weather station data from 6 airports, starred on this map, from 2000 to 2019. These stations recorded average monthly temperatures. From the graph above, there doesn't seem to be a noticeable trend in temperature over this 20-year time span, although the slope of the line of best fit is positive. They also recorded total monthly precipitation data in inches, for which there is a definite increasing trend, matching our predictions that winter precipitation and storms will increase. Lastly, drought data came from the U.S. Drought Monitor, which categorized percentages of land data in Michigan by drought severity. There seems to be a downward trend, which contradicts the drought predictions from the climate projections. This should be looked into with further research. I tried to find the monthly West Nile virus surveillance data, but there was none available, so I had to adjust the other variables to annual values. Finding data was a struggle because I needed it to be specific enough to accurately represent a single state. I removed data from 2000 and 2001, as there were no cases until 2002, which experienced a drastic peak from a lack of immunity, something not likely to happen again. Lastly, I removed West Nile case statistical outliers in 2005 and 2018. This table contains the data actually used to make the model. I conducted multiple linear regression, and the R-squared value is shown to be 0.72. This means that approximately 72% of the variance of West Nile cases can be accounted for by my model. Additionally, temperature seemed to be the strongest predictor of West Nile, with a p-value of 0.0014, making it significant, and drought seemed to be the weakest predictor, with a p-value of 0.59, making it insignificant. You can see a visible difference between them, with the blue being actual West Nile cases and the red being the predicted values.
Hi, my name is Yarianis, and my partner is Yariel, and we are part of NASA's Interns 2020 Mosquito Mappers Research Group.
Our project is investigation of Puerto Rico communities focused on mosquito habitats and awareness.
The abstract is the massive spread that mosquitoes are having in Puerto Rico. Is why we want to investigate in detail about Puerto Rico's community focus on mosquito habitats and awareness. Throughout the experiments, mosquito habitats were found in the mosquito species Aedes aegypti and Culex, some imposters such as ants and midge larvae Clinotanibus. We considered that one of the factors that attracted mosquitoes was the natural CO2. At the same time, the population is aware of eliminating these breeding mosquito habitats.
Mosquitoes habitats influence the massive spread of mosquitoes. If the climate of Puerto Rico is a warm, humid, tropical climate with rainfall throughout the year, mosquitoes will be captured and observed in the larvae mosquito traps. Since Puerto Rico is an island with high mosquito increases, the majority of Puerto Ricans will have knowledge and awareness about mosquitoes.
Introduction and our desire is to provide information with proof to Puerto Ricans, and not only them, but also those who are able to read our information. The problem of our research is the massive spread that mosquitoes are having. It's important that Puerto Ricans are aware of mosquitoes since the island has a large number of mosquitoes due to its climate that favors mosquito average.
We used 4 methods. One of them was the mosquito larvae traps. This was used to obviously trap the larvae and mosquito we are investigating. The collect earth was used to analyze the land sites of the places we are investigating the mosquitoes. The global serve was used to identify the mosquito habitats and the larvae. And for last, it was the Google Forms that was used to the online survey.
The results of Collect Earth Online was on Manatee land site and Vega Baja land site. We analyzed the land sites, indicating if it had trees, grass, buildings, water sources, or even shadows. This is the table results of mosquito larvae trap. We divided the table in temperatures, monthly precipitation, the location, if the traps had mosquito egg pupa larvae presence, if it had imposters, and what was the bait type. This was the table used in the experiment 1 of results of mosquito larvae trap. The only difference here is that this has quantity of mosquitoes since the first didn't have mosquitoes trapped, and the type of mosquito found. We identify the location and MUC description of the land covers. In the mosquito habitat mapper section, we identify the mosquito habitat, water source, mosquito egg pupa larvae presence, the quantity of mosquito, and the type of mosquito found.
Google Forms online survey. The survey was used for the purpose of collect data from residents of Puerto Rico. A total of 219 residents of Puerto Rico answered the survey. Here we see some examples of graphics of the answer of the Puerto Ricans.
We obtained a correct hypothesis and a survey. In addition, we now have the knowledge of Puerto Ricans, knowing and having awareness of mosquitoes.